Well, just when we let him in, somebody's gonna talk. I'll wait until everyone until it stops before I start talking. Uh-oh.
Julie, you will start?
Oh. Be getting started in just a couple minutes. And we give another minute to let everybody come in. A few more seconds. Okay, I know we're expecting some more, but I think we'll get started.
How many people are there?
Hmm?
How many attendees
We're
are
gonna
there?
get started. Oh, don't remember, over 20,
Good.
over 20, 25, something like that, signed up for tonight. Yeah. So we'll get started though, because maybe they're gonna come in later. So anyway, hi everyone. I'm Julie Gertze, an RN and founder of RAN New England. You may hear us refer to our organization as RNE. RNE is a nonprofit organization devoted to bringing education and information about rare diseases to all who touch the rare disease community. Welcome to RNE's Career Fair and Medical Genetics. We are grateful to everyone for being here tonight. When we hear the word rare, we think of a small number. But in reality, there are collectively 30 million people diagnosed in the United States and 350 million affected globally by a rare disease. Statistically, 80% of these rare disease patients have a genetic disorder. There are not enough geneticists to care for these patients, and this situation will only get worse as technology moves forward and patients are better and faster diagnosed. Remember, these patients are sick and may not be receiving appropriate help while they wait. They are hurting and sometimes dying because others just don't understand what is wrong with them. Something must be done to help these families, and RNE is trying to make an effort in this area. Let me now introduce you to our host for tonight's event, Dr. Mark Corson. Dr. Corson is a pediatrician who specializes in metabolic genetics. He has directed the metabolic clinic at Children's Hospital and Tufts Medical Center in Boston. He is co-founder and co-director of the North American Metabolic Academy. Currently, he is director of the Physician Support Service and director of education at VMP Genetics. He is also a founding board member of RAN New England. A more complete biograph can be found in the Career Fair brochure sent to you. Through RNE, Dr. Corson has directed informational opportunities such as this career fair tonight in order to make a positive impact for all genetic disease stakeholders. Mark, take it away.
Thank you, Julie. And thanks everybody for attending Rare New England's Genetics Career Fair. I want to thank the following sponsors for their generous support of the fairs. One, the New England Regional Genetics Network, Homology Medicines, Ionis Pharmaceuticals, Ultragenyx Pharmaceutical, and Vertex Pharmaceuticals. Again, as Julie said, my specialty is pediatrics with a focus on genetics as a subspecialty, specifically the area of biochemical or metabolic genetics. So if you think back on your biochemistry, I deal with patients and families who have defects in those biochemical pathways. Now, biochemistry can be abstract and rather dry, but it's that patient community that really puts the human back in biochemistry and shows how relevant the urea cycle or the electron transport chain really is. Why are we doing this program tonight? Well, genetics impacts almost every area of medicine. So as you can imagine, there's a growing need for geneticists in this country and abroad. Patients and families want and need to hear information about the diseases they have, information to plan their futures, information as a couple with a family history looking to have children, and information about effective therapies now available for genetic disorders. And considering that there are over 7,000 rare diseases, not all genetic, and growing, that's a lot of information that needs to be shared. But the impact of genetics is not just in rare diseases. Genetics can impact how we manifest lots of mainstream diseases, including COVID, how we respond to the disease, how we utilize medications, what medications are available or effective or safe, and so on. This evening, you will hear from 3 physicians who specialize in the area of genetics: a general clinical geneticist, a specialist who treats patients with genetic metabolic diseases, and a laboratory geneticist. You'll hear a bit about what they are doing, how their genetics expertise has helped to treat their patients and families, and how the experience has impacted their own lives. A Q&A session will follow the 3 presentations. Please submit your questions as we go in the Q&A area. After that, I will guide you through additional opportunities to learn more about genetics. But first, you might think that liking biochemistry makes me a nerd, and the fact that I bring some biochemistry into the clinic makes me even more of a nerd. Well, fun fact, I am a nerd, but we genetics nerds can have fun. Let me show you this very interesting genetics pedigree. Many, many years ago, when I was 23, I was married to a widow who was pretty as can be. This widow had a grown-up daughter who had hair of red. My father fell in love with her, and soon they two were wed. This made my dad my son-in-law and really changed my life. Now my daughter was my mother because she was my father's wife. To complicate the matter, even though it brought me joy, I soon became the father of a bouncing baby boy. My little baby then became a brother-in-law to dad, and so became my uncle, though it made me very sad. For if he was my uncle, then that also made him brother of the widow's grown-up daughter, who of course was my stepmother. Father's wife then had a son who kept him on the run, and he became my grandchild, for he was my daughter's son. My wife is now my mother's mother, and it makes me blue, because although she is my wife, she's my grandmother too. Now, if my wife is my grandmother, then I'm her grandchild. And every time I think of it, it nearly drives me wild, because now I have become the strangest case you ever saw. As husband of my grandmother, I am my own grandpa. Oh, I'm my own grandpa. I'm my own grandpa. It sounds funny, I know, but it really is so. I'm my own grandpa. Written back in 1948, you'll find the YouTube is full of people singing that song. But I'm the first one actually to ever actually drafted the pedigree. So that is one of, I guess, my claim to fame. So let's begin. I'd like to introduce to you first Dr. Jody Hoffman. Dr. Hoffman is Associate Professor of Pediatrics at Boston University School of Medicine and the Chief of the Division of Genetics at Boston Medical Center. She also serves as Medical Director of the Boston University Genetic Counseling Master's Program. She received her medical degree at Albert Einstein in the Bronx. She completed a pediatric residency at Yale Children's Hospital in Connecticut and her genetics training was completed at Children's Hospital of Philadelphia. She practices as a general medical geneticist and provides counseling, diagnostic care, and management regarding prenatal care, newborns, children, and adults with genetic conditions. She plays a role in the optimization of lab utilization of genetic testing, serves as a member of the Scientific Advisory Council for the National Tay-Sachs and Allied Diseases Organization, and aids in the release of incidental genetic findings for the Framingham Heart Study. For more information about Dr. Hoffman, please see her bio in the Career Fair brochure. Thank you, Jody, for joining us. Jody, your microphone is off. Thank
There
you.
we go. Okay, thank you for that introduction, Dr. Corson. And so everybody should know, I was lucky enough to first meet Dr. Corson in about, I'm going to say 1992, when he took me to a summer program for teenagers with PKU. And I was already in love with genetics, but that summer with those lovely teenage girls who really needed to learn a lot more about how to keep themselves healthy was definitely a strong influence in my life. And I hope you'll all have such a experience to inspire you. So I was asked today to talk a little bit about my interest in genetics and, what I do, what interests me, and what makes it exciting. So we'll talk a little bit about my experiences. First of all, I love genetics because at a deep level, the genetics is what makes our bodies form the way they are and work as perfectly as they do. And I think every specialist out there thinks my organ of the body is most important. But honestly, with all due respect to neurosurgeons and cardiologists, there would be no brain or no heart without our genes. So it's really our genes that are controlling our entire health and function of the body. And it's absolutely fascinating and amazing to me that so often everything works perfectly well. But we are in the situation of taking care of those who have the misfortune of having a small difference in how their body was formed and feel concerned about it. So we have people come to us for all sorts of reasons. Sometimes the outsides of their bodies are formed differently, sometimes the insides of their bodies, sometimes their skin. Sometimes the way that their connections are made in their brain are different than other people. Some people have unusual behaviors or seizures with other features. And then we have a lot of people who don't necessarily even currently have a condition, but may have a family history of certain conditions, may have cancer or family history of cancer that leads them to think they may be predisposed. And all of these folks may hope that we can give them some more information about how to take care of themselves, keep themselves and their family members healthy, and really optimize their quality of life. So one of my favorite patients that I saw in the last few years was a young lady. She's 8 years old, of African-American background, and she had not been seen in genetics clinic for, I would say, 3 to 5 years. And her mother came to me in order to see if there were any updates and helping understand what was making her have several medical issues, including seizures, the inability to speak, and the inability to walk. So she was a very happy child sitting in a wheelchair, communicating with her hands and her smiles, but she was unable to interact the way that many other people do. She had had extensive testing. All sorts of genes were tested for, chromosomes, pieces of chromosomes, and it had been quite a while. And since that time, a new panel had been developed of genes that could look at about 250 different genes at once that predispose people to seizures, sometimes just seizures, and sometimes seizures with other issues like intellectual differences or difficulties with gait. So we sent off a blood sample, just a teaspoon of blood, off to a genetic testing company, and back came some results that said, wow, this child actually has a specific pathogenic variant, an abnormality in the code of the gene that causes a condition called Mowat-Wilson syndrome. And I said to myself, gee, I know what Mowat-Wilson syndrome is, and I'm kind of disappointed that I didn't think of this syndrome on my own. But given that, as Dr. Corson said, there are many, many different conditions and 22,000 genes, we do give ourselves a little bit of leeway to not know everything all the time. But I do think that in this case, I had a patient with Mowat-Wilson syndrome before, but that child was of a Caucasian background. And this child was of an African American background. And it's quite interesting that much of the literature out there and many of the textbooks have pictures only of children with white skin. And I also think that there are some differences in structure, overall structure of some facial features in some diverse backgrounds that can make it harder for geneticists to look at a person and say, you have this condition if we don't have a variety of patients pictured in our literature. So this patient really brought to mind that I need to think a little bit more broadly about the way that I learn and think about genetic conditions, especially those that have facial features that we try to recognize. So here's a classic series of pictures of a child who has Mowat-Wilson syndrome, and you see he is, of course, Caucasian. And this mother actually said to me, hey, doc, like, why do you think it took us so long to diagnose this condition? I said, well, one is that we didn't have the seizure panel available, but the other is that unfortunately, a lot of us see only pictures over and over again of kids with a specific facial background. And so you can see this little boy here. He has, facial features that become more prominent with time. And here's my patient here who, was an adorable little girl who maybe if I looked very, very carefully, I could see that her earlobes pushed up a little bit, which is a symptom that we can see in Mowat-Wilson syndrome. But it really wasn't so obvious to me. She looked a lot like her family, and she didn't look as much like the pictures of the literature as we've seen before. But this family was extremely grateful for this diagnosis. After 8 years of not knowing what was going on with the child, we were able to connect them to support groups. And actually, my previous patient I had diagnosed with Mowat-Wilson syndrome, and the girls have gotten together and interacted. And this mother has been able to understand now the why behind this condition. It was sporadic, new to the child. And that it's unlikely to happen again in the family, and also what to expect from this other family who also has a child with this condition, as well as what we could share from the literature. So I think it was quite rewarding to the family to provide a diagnosis after this long odyssey. There are other people who I meet who already have a diagnosis. So I met a young lady when I worked at a clinic in Fall River, and she had a skeletal dysplasia, which is a disorder of the way that the bones are formed, called pycnodysostosis. So she was quite short. Her fingers were very short. She had kind of extra creases along the back of her fingers. Her mid-facial region was a bit depressed like these children. This is a young man, actually, and also a somewhat prominent nose. And she was born to teenage parents who were about 15 and 16 at the time of her birth. And they became extraordinarily wonderful advocates for this child and did everything they possibly could in life to help her succeed. They stayed together as a couple. They grew up together. They took care of this child. And I got to see different milestones in her life. When she learned to drive, she had to have extensions on the pedals of her car. I got to write a recommendation for her to go to University of Pennsylvania, where she thrived and met lots of different political figures. And now she's in, and actually she gave some advice to me about sending my daughter to college. And now she's actually at a school of public health in New York. And I know that she's going to do wonderful things. So, we have long-term relationships with some of our patients, and they fight such difficult battles to get where they need to get in life, but they have such strength that it's extremely inspiring. Then I have a young boy who presented to me at about, I would say, 10 months when I was at Tufts Medical Center after Dr. Corson had briefly after he had left. And this child had some coarse facial features, which can be a sign of some storage material in the face, a buildup of some proteins from enzymes that aren't working, a gibbous deformity, which is a prominence of the lower portion of the back, chronic mucus flowing down through his nose and throat. He ended up needing his tonsils and adenoids removed at 9 months, which is almost unheard of. But to me, that combination of those three things automatically pointed me towards a storage disorder. And so we were able to get him diagnosed with a storage disorder in a very short time. He was found to have a condition called Hunter syndrome. And this is a lysosomal storage disease for which there is actually a treatment of bone marrow transplantation if it occurs early on in age. So he actually had a bone marrow transplant. And as you can see here, he's about 4 or 5 years old in these pictures. He is one schnazzy, sharp kid who's doing tremendously well. And had we not been able to recognize what this condition was and get him treated in time, things may have turned out quite differently. And so some of the things that Dr. Corson mentioned, I enjoy in addition to my clinical time is participating in research. So I once had a child who had the lack of formation of the nose, and I was able to submit that child's sample to a local group that was looking at translocations, which is when chromosomes split, break, and attach again in a different formation than they're supposed to. And it turned out that that gene actually disrupted formation of the nose. And they were able to create a zebrafish model that showed that if you mutate that specific gene, that the nasal portion of the zebrafish would not form properly. And so really taking, bench to bedside and bedside to bench in terms of research and going back and forth is very exciting to me. I also really enjoy working with certain groups like the National Tay-Sachs group and helping them optimize testing for children in terms of preconception screening or prenatal screening, or even once a child is born. And I also feel that it's very important to act as an advocate for some of our patients when it comes to court cases that come up and situations that have arisen, such as children that have fractures and bruising, which may be secondary to a genetic syndrome as opposed to abuse per se. And then I very much enjoy the opportunity to educate genetic counseling students in particular who are at Boston University, as well as previously when I was at Tufts and we had fellows in genetics who officially are residents, but people who had completed their residency and then went on to do a subspecialization in genetics. I very much enjoy educating those people who are interested in the same types of things that I'm interested in. And, we really enjoy working and learning together. So for all of these reasons, I've been very grateful for my career in genetics. And of course, to Dr. Corson, who was one of the people that influenced me in my career. And I hope that you will continue to think about a career in genetics, and I'll be around for questions at the end.
Thank you, Jody. Thank you very much. And again, put your questions in the Q&A box, and I'll address them to the panelists after their presentations. On to Dr. David Weinstein. Dr. Weinstein graduated from Connecticut's Trinity College and earned his medical degree at Harvard Medical School. At Boston Children's Hospital, he completed his residency, chief residency, and fellowship in pediatric endocrinology, and completed his master's in clinical investigation at Harvard and MIT. He became the director of the Glycogen Storage Disease Program at Children's before moving to Florida in 2005 to pursue gene therapy on dogs with naturally occurring GSD. He then went on to serve as professor in the Department of Pediatrics at University of Connecticut School of Medicine and director of their Glycogen Storage Disease Program, a joint venture of UConn Health and Connecticut Children's. Dr. Weinstein specializes in disorders of carbohydrate metabolism with a particular focus on GSD, glycogen storage disease, but also hypoglycemia and diabetes. He managed the inpatient and outpatient care for the largest cohort of GSD patients in the world, following over 500 pediatric and adult patients with this disorder. These patients came from over 49 states and 43 countries to be cared for him. It was the largest clinical and research program of its kind in the world. In addition to local GSD community outreach, his team partnered internationally with institutions and scientists in Argentina, Brazil, Canada, China, the Faroe Islands, Israel, Mexico, and the Netherlands. As a physician scientist, he has authored over 80 articles and 26 textbook chapters on GSD. But notably, he's the recipient of the prestigious International Humanitarian Award, the Order of the Smile, for helping children around the world with glycogen storage disease, a recognition shared with Pope John Paul II, Mother Teresa, Nelson Mandela, and Pope Francis. He was named one of the inaugural Goldwater Scholars in 1989. He is a former Jan Albrecht Award winner from the American Association for the Study of Liver Diseases, and received the George Satcher Award from the Gerontological Society of America. He was inducted into the Rare Disease Research Hall of Fame in 2013. He is now involved in participating in human safety trials of his gene therapy. For more information about Dr. Weinstein, please see his bio in the career fair brochure. David, thank you for joining us.
So thank you, Mark. So my confession, first of all, is that I'm a nerd, and except I won't be singing tonight. I think that would not be a pretty picture. But I want to thank you. I want to thank Rare New England, but I also want to thank you personally, because you were one of the reasons I went into genetics metabolism. You inspired me, and it was the best decision that I made. And I hope today to be able to convince some of the participants to follow my path. So give me 1 second. I'm just going to, so can you see the screen? Are people able to see the screen?
Yes.
Okay, perfect, thank you. So I'm calling this Living a Life That Matters, Lessons from a Rare Disease. And I'm gonna take you back to when I was in the same situation where many of you are right now, trying to figure out what I was going to do with my life. And I went to speak to my mentor about the struggle I was having and trying to figure out what area of medicine to go into. And he gave me a book. He wouldn't give me advice. He said, take this book and read it. And the book is called Living a Life That Matters. And it's about balancing what is a success? Is it money? Is it titles? Is it publications? With what makes you happy? And the book has a theme, which is that all people can make a difference. You just have to find the area that makes you happy. And I'm showing you this because this book changed the way I looked at medicine and ultimately led to me going into genetics. So I began my career as an Alzheimer's pharmacologist. In 1989, I moved to Boston to pursue Alzheimer's research. I had been doing research in Alzheimer's disease for several years, and I was a young, idealistic person, and I decided I was going to move to Boston because 3 of the 5 biggest researchers in Alzheimer's disease were in Boston. Fortunately, the Massachusetts General Hospital gave me a lab space, which had been a closet before I moved into it. But I was thrilled just to have that. And I thought this was going to be the greatest experience for me. Fortunately, I had success. And as you heard, in 1991, I was honored with the recognition of having the best research in the field of gerontology by a graduate student in the country. But what I realized is that even though the research was successful, I wasn't happy. And I wasn't happy because I wasn't making a difference in patients. The research was too far away from the patients. And even though it was going well, I wanted to see something that was going to make a difference. I also was turned off by the fact that in these big diseases like Alzheimer's disease, people didn't work together. When I was shocked to see that even though all these great researchers were at the same institution, nobody spoke to each other. In fact, people would lock their lab books, their notebooks up in a safe at night to keep other people from seeing it. It was crazy. And I decided at that point that even though research was going well, I wasn't thrilled. I also didn't enjoy watching people deteriorate. I made a decision that my mentors thought was crazy, which is I went from gerontology to pediatrics. And I was probably the only person who's ever been a member of the Gerontological Society of America and the American Academy of Pediatrics at the same time. Well, I went to Boston Children's, that's where I met Mark, and I became a pediatric endocrinologist. And while I was a fellow, I was studying hypoglycemia with Mark. And in 1998, I got invited to the Association for Glycogen Storage Disease Conference. I asked them, why are you inviting me? I only have seen 2 patients with glycogen storage disease. And they said to me, well, first of all, we invited your mentors, but they didn't want to come. And they said, you take good care of your patients. And I said, but I've only seen 2 people. They said, doesn't matter, with 2 people, you're an expert. So I decided, okay, it was a free trip to Seattle. I really did not have interest in glycogen storage disease, if I tell you the truth. I went there because I wanted to see Seattle. And I got to this conference and I was shocked by what I saw. The conference began with a moment of silence, and they read the names of all the children in the United States and Canada who had died that year from glycogen storage disease. The conference was devoted completely to liver transplantation. There was no new research. In fact, the research that was presented was 16 years old. And if there's no new research, there's no hope for things getting better. Well, I sat down at a table with, I didn't know anybody there, and I sat down at a table with a lady who was sitting by herself. And she started talking to her, and she told me about her twin boys. They were 1 year of age, and she just hoped that they would get a liver transplanted before they died. And I think you probably realize a liver transplant in a 1 year old is not a great solution. The other thing she told me is that she had twins, and she didn't know what was going to happen when she got called that they had an organ for one person. How do you choose one of your children to live and one of your children not to live? I had a son who was 1 month older, 1 month younger than her boys, and it hit home. I said, nobody should ever be in this situation. And these children deserve better. And I went back to Boston Children's, and I told my division chief, the world doesn't need another diabetes doctor. I'm going to study glycogen storage disease. And he told me I had flipped out, that I was crazy. He told me I was, quote, committing academic suicide, that there was no need for a glycogen storage disease doctor. There were no grants. In fact, there was only $7,500 worth of grants every year for GSD. But I followed my heart instead of my brain. If I thought about it, I never would have done it. But I had met Mark, and I decided, you know what? Genetic disease is where I can make a difference. And I started doing GSD work. And this is not going to be a glycogen storage disease talk, but with advances in treatment, patients became healthy. And you look at this slide here, you would never know anybody had glycogen storage disease if they had a disorder that was once considered fatal. What you can see there are people doing every sports, every activity. Nobody had ever had a child with where the mother had GSD until 2001. And by the time I left clinical practice a few months ago, we had had 58 children born to mothers in the program. And one of the perks, by the way, for people who go into genetic diseases, you really get to know your family as well. And I had many patients that I followed for 25 years continuously. And when they have babies, you're part of the family. And one of the biggest honors for me was being able to be at the hospital at the parents' request to be there when the baby was born. And several of the children are named David. That's also a big honor. It's really just a family. The biggest honor for me was that 7 of my former patients became doctors, and they wanted to pursue this, and 2 of them came to work with me. And for me to see it progress to that level was absolutely incredible. And to be able to work with my former patients was a dream. But ultimately I wanted better. So I've been pursuing gene therapy since 1998. It started in the lab in liver cells, then went to mice. And then we did, as Mark said, gene therapy in the naturally occurring dogs, and it worked. And the culmination of my academic career occurred in July 2018, when I had the opportunity to treat one of my patients with gene therapy, and ultimately 12 of the patients have been treated now. This patient is now almost 3 years out, and he is completely off his cornstarch treatment. He's not on any medical treatment. And in the past, that would have been fatal. So to see the progression from a fatal disease to one where we've been able to do gene therapy and people are doing well is just worth more than any money. A few months ago, I made the transition to industry. With COVID, things changed. I'm not really going to go into that because we don't have time, but my goal now is to bring gene therapy to other rare disease populations. So we are starting this month gene therapy for GM1 gangliosidosis, another rare disorder, which is a neurologic disorder where kids deteriorate. And I also want to thank Jody for her work on NTSAD, which is one of the foundations that is very involved with helping these children. But I'm very optimistic for the future. But the take-home message is that we need more help. Bigger is not always better. You can make a difference in genetics, unlike the fields of diabetes and Alzheimer's and things like that. You can make a huge impact in the field of genetics. These patients, as you just heard from Jody, these patients suffer because doctors don't know these disorders, and the kids deserve better. I encourage people to take chances. Going into genetics is not always the safest way because it's a struggle to try to make it, but it's the most rewarding decision you'll ever make. And don't lose your idealism. You're going into medicine, or you went into medicine to help people, and this is really how you can do it. So if anybody wants to talk about industry versus academics, we can talk about that later. But thanks for giving me the opportunity to talk to you.
Thank you so much, David. It's great to hear about your journey. All right, our final speaker, Dr. Lisa Braley. She obtained her undergraduate degree in biology from Harvard University and her medical degree from the University of Connecticut. She completed residency and fellowship training in genetics at Yale and is board certified in clinical genetics, cytogenetics, and molecular genetics. She is currently the Associate Scientific Director of Cytogenetics for H&L Lab Medicine in Allentown, Pennsylvania, and serves on the board of directors of the New England Regional Genetics Group. She's also served as Associate Director of Cytogenetics at Ameripath Northeast of Quest Diagnostics and as Technical Director of Molecular Diagnostics and Clinical Trials at Dynon Systems, both in Shelton, Connecticut. And she has been an adjunct faculty member at the University of Connecticut and University of New Haven. Her interests include DNA sequencing and data analysis, genetics education and outreach, and metabolic disease. She and her husband reside in Derby, Connecticut, where she enjoys hiking, swimming, playing the violin, reading, and promoting community engagement. For more information about Dr. Braley, please see her bio in the career fair brochure. Welcome, Lisa. Turn on your mic. Glad you're here.
Thank you so much for having me. And again, thank you for the introduction. I must say, I always feel so fortunate to have colleagues like you, David, Jody. Geneticists are a really interesting and dynamic group, and it's really fun to be around you all. So again, I'm Lisa Braley. I'm a medical doctor and geneticist, currently working at a cytogenetics lab in Allentown, Pennsylvania. I graduated from medical school in 1998, and I really wasn't sure what I wanted to specialize in. I was a little older student. I'd had a little delay getting to medical school. And as an older student, I realized I really didn't like sleep deprivation, being on call. So I was really torn about going into patient care. I thought I wanted to be a pediatrician, but that call scared me. My favorite coursework in medical school was my genetics class. I loved the logic of DNA, the elegance of how DNA works. In high school and in summers in college, I had worked in group homes for the developmentally disabled and had really seen almost like a textbook worth of different syndromes. And back then, most of them did not have laboratory diagnoses, they had clinical diagnoses. And it's been really exciting to me over the years to see almost all of those conditions wind up with a laboratory diagnostic as we have learned more about DNA and developed bigger and better testing methods. So from this point of view of having wound up in the laboratory, I'd really like to emphasize that there are many pathways through a medical career. Not everybody winds up seeing patients. Like probably most people, when they interviewed and when they thought about being a doctor, were thinking about patient care. But many people in medicine wind up in administration, research, insurance companies, big pharma, learning gene therapy. So there are numerous ways to be in medicine within sort of more traditional medical fields. Things like pathology, radiology, lab medicine, and laboratory genetics are very real career options. And I want to, in the vein of, like Dr. Weinstein talked about, of go where you're happy. One does not need to feel obliged to be in patient care, to be a useful physician. So all of these choices are legitimate. And I made my choice for both positive and negative reasons. By positive reasons, I mean love of a field. DNA is cool. The people I saw around me in genetics were really interesting, diverse, came out of so many different areas. DNA affects everything. Some of my reasons are negative. I couldn't deal with that sleep issue. I really wanted a place where I had better control over my day. And I think that's important to say, that it's okay to think about what kind of lifestyle you want. Particularly as an older student, it was important to me to have more control over my time. Not that it's a problem if people want to be on call, but so I ended up I am triple board certified in genetics. I did clinical genetics, cytogenetics, and molecular genetics, and have focused on the laboratory options there. These days, one cannot actually go into cytogenetics or molecular genetics separately. They have been combined by the American Board of Medical Genetics into laboratory genetics and genomics, or LGG. And I think that's been a good change, because it means that people are trained in the broad range of genetic testing methodologies, not just looking at chromosomes or just looking at DNA, looking at it all, the genome. So let me just find, I'm deviating from my script, I have to find where I am. So another point that I would make is both PhDs and MDs can certify in laboratory genetics and genomics. So I don't have to be an MD to do what I'm doing. I am glad that I am. Having the medical background, so having seen patients, being board certified in clinical genetics, actually evaluating patients, though that's not what I do now, gives me a perspective on what I do. Being trained in the spectrum of medicine, though again, it's not my practice, means that when I approach a case, I can tell that I have understanding that a PhD doesn't always have. I have vocabulary from medical school. So I think I come at it with somewhat of an advantage from the background that's not necessary. It's also important to realize that it is possible to work in a lab and also to see patients. It would be completely reasonable to be at, say, an academic medical center and to have clinic a couple days a week and to run a lab. So the fact that I'm an MD in a laboratory is a logical choice for me. And certainly as that situation can be put to different use in different types of settings. I'm at a commercial lab. So back to my love of DNA. I've always felt like DNA isn't one of the simplest things in the world. Four building blocks, they match up. Pretty much that explains almost everything about DNA. Super simple, except that there's always a twist. There's a double helix in everything. So there's always something different. So the basis of everything is those two strands line up in opposite directions. Those bases, the building blocks match up, but there's imprinting. There's always a different way that DNA can fool you. So I'm going to tell you two simple stories. I'm sorry I don't have slides. I hope that my words will convey my stories. So one of my stories is about a life-altering genetic diagnosis. One is about a fairly simple diagnosis that had a twist in it that sort of obscured what the diagnosis was. So case one is a 10-year-old girl who was small for her age and had mild cognitive impairment. Not a lot going on until in the previous year, she had started developing seizures that were refractory to treatment and seemed to be losing cognitive milestones. So she's suddenly going backwards. There's regression. Her parents had been divorced for several years, and a major issue in their relationship over this girl's life had been the father's accusation that the daughter had problems because of fetal alcohol syndrome. So he was accusing the mother of drinking. Maybe she had drunk some, but the mother denied that there had been any amount of drinking that would have caused this. The girl had never had any genetic evaluation because her findings until now had been fairly mild. So she was seen at the Yale Child Study Center, and a chromosome evaluation was sent. These stories both go back several years, but have a laboratory basis to them that I thought was relevant. So a chromosome analysis was sent. That was the standard at the time. There was no whole genome sequencing, next generation sequencing. The microarray wasn't available. The chromosome evaluation showed that she had a ring chromosome 20. She had the normal she had 46 chromosomes, but one of her chromosomes 20 had popped in on itself and formed into a ring. All of the chromosome material seemed to be present. So it turns out that ring 20 is a cause of intractable seizures. So the rings are unstable. They can sometimes, when the cells try to replicate their DNA, the ring will sometimes get stuck on itself. And that can, when the cell tries to divide, that can break apart and the cell can lose the chromosome, or sometimes those chromosomes can pop up and become a double ring or even a triple ring. So the cells have different chromosome complements. Leaving a child mosaic for loss of chromosome 20, the ring, or extra chromosome 20. And a feature of ring 20 syndrome is intractable seizures. So what we had there was a perfect explanation for the phenotype of the child that we had in front of us. For probably no influence of fetal alcohol syndrome, whether or not the mother had drunk anything during her pregnancy, that was not an impact on this child's situation. So we were able to give a complete explanation that saved this mother from a lawsuit, could potentially have saved this family from strife over the years if they had known this earlier. So it was very cool to me to help this woman with her husband, ex-husband, to have the child have a diagnosis that made sense. Not necessarily enable a lot of treatment. You can't fix that ring, but at least you can know what has worked best in the past for ring chromosome 20 syndrome. And you really wonder whether their life trajectory could have been different had they recognized this as a syndrome and had genetic testing earlier. So my second case is one with a little twist to it. In this one, I was called to, again, in this instance, I was the clinical geneticist, a resident, and we were called to see a baby in the NICU, and that baby meowed. So she also had cardiac defect and a lot of other things, but when she cried, she had a meow cry, which is a known distinctive finding of cri du chat, or cat's cry syndrome, which is due to a loss, typically due to a loss of material from the short arm of chromosome 5. So the NICU knew this. They were smart. They knew what the meow would mean. They had sent chromosomes and there was nothing missing from chromosome 5. The chromosomes were completely normal. So I was there and said, oh my gosh, this baby meows. She has to have Cri-du-Chat syndrome. But it was being dismissed as a diagnosis because the chromosome 5 looked fine. So in discussion with the lab, there was, should we do any further testing for chromosome 5? What should we do here? We got out a FISH probe, fluorescence in situ hybridization, for the short arm of chromosome 5, and it turned out that this probe did not hybridize, so there was something missing, but it sure didn't look like it. Usually you can see the loss of this piece of chromosome 5 under the microscope, but this, it didn't look like it was there. There was any problem, but it was missing with the fluorescent probe. So what was happening there turned out to be another test, which was called subtelomeric FISH, which showed that there was a piece of chromosome 14 that got stuck on chromosome 5, a translocation, like Dr. Hoffman, Jody talked about, and that the end of chromosome 14 looks virtually identical to the top portion of chromosome 5, and so it just replaced it. So this baby actually had extra chromosome 14 and missing chromosome 5, so really did have Cri-du-Chat syndrome, and also had additional syndromic features, the cardiac defects, because of having the extra material from chromosome 14. What I loved about this was that the clinical findings mattered. Like if you stood at that bedside and you snapped the bottom of that baby's foot and heard that baby go, wow, there's no way you could think anything other than this baby had Cri-du-Chat, but you would be walked away from that diagnosis by the chromosome findings, and you had to be persistent enough to find it. Now, nowadays, the first test that would be sent would probably be a microarray, and a microarray would find that immediately. So this situation would be different now. And that's another thing I love about genetics. There's new testing. There's new ways to look at it. And a microarray, I'm going to throw in a little point, microarray might not have found the ring 20 from the first child because there's nothing missing necessarily. That ring just forms and there's not necessarily any material not there. So a microarray would be the perfect test for my Cri-du-Chat patient and might not have found my ring 20 patient at all. So I'll be done in 2 seconds. So I love the variety of what one can see, the fact that you can change lives, the great colleagues that I have, and the dynamic changing field of genetics.
Thank you, Lisa. And it's very important to make mention that, you know, the clinicians and the laboratory folks in genetics work extraordinarily close with each other. And so both benefit from the expertise of the other. So thank you very much, Lisa. At this point, there are 2 things. We're going to do the Q&A and address the questions that have come in. If others have questions, please write them in. And then I want to finish by giving you additional opportunities if you want to consider genetics, what additional opportunities are available through R&E, but also out in the genetics community. So first question, Jody, how involved are you with genetic counselors? If you are, what kind of work do you do with them?
So in my practice, we have a pretty small practice. So I partner closely with a genetic counselor, and we happen to work in parallel, seeing 2 patients at the same time. And she takes a lot of time speaking to her patient, gathering information, reviewing records, explaining possible testing. And then I tend to join the counselor to do a physical exam, to consider whether the plan might shift based on that physical exam. and then telling the patient together what our thoughts are and what the next steps are. And then I also partner with 2 genetic counselors in our antenatal unit, which is where we take care of pregnant women and pregnant people, as well as our cancer center, where we have 2 genetic counselors who discuss more complex cases with me, get feedback, and we partner together to make sure that their patients are all well taken care of. And then, of course, in co-educating all of our students in the master's program. So we do spend a lot of time together and try to bring our various skill sets to taking care of our patients.
Thank you, Jody. David, from an attendee, I have a particular interest in CRISPR and neurodegenerative disorders like Parkinson's. Can you give your professional opinion on the promise that you see in gene editing as compared to gene therapy, particularly in regards to in vivo treatments of adults?
Sure. Not an easy question to answer quickly, but I think the future is bright for both, and there's a role for both gene therapy and gene editing. Gene therapy is finally showing the promise after hearing about this for 20 to 30 years. I remember in 1999, Time ran a cover saying that it was the year of gene therapy. And of course, it took decades more to get to it. But we're seeing now that gene therapy is working and it's having success. But gene therapy is not a cure. We know that in many organs, it's not going to last forever. And so there is a role for gene editing. And I think CRISPR, and I'm even a bigger fan of the next generation of CRISPR called where it's gene editing, base editing, where it's just one base that's changed that some of the companies are working on. Because I think there's less likely the potential for damage. The biggest limitation to CRISPR or to the base editing is going to be the fact that they are individualized, and it works well when you have one common mutation. But if you have a rare disease and there are many different mutations, the companies are not going to make these treatments for every mutation, especially ones that there are only 1 or 2 children with. So I see a role for both gene editing in the future, which would be a permanent correction, and gene therapy, which is showing the potential. And then if I have 1 more minute, for the neurologic and neurodegenerative disorders, this is a very exciting time. I know that there are trials going on now for gene therapy for Parkinson's. At our company, at Passage Bio, we're starting FTD, which is another form of dementia. The trials are starting in the next month or 2. We never thought you could treat a neurologic disorder, at least when I was doing a disorder like that, when I was doing Alzheimer's work. And I think the future is very bright. The challenge is, how do you get it to the brain? Can you do it through delivery with a vector, a virus like AAV9, or do you give it by injecting directly into the CSF? I think that's the challenge right now on how to do the gene repair or the gene therapy for the brain. But these are exciting times.
Excellent, thank you. Lisa, thank you for your honest and realistic view of lifestyle as a physician. When in genetics training do you have to declare yourself in terms of clinician, research, or laboratory?
One could declare oneself right up front. There's some different avenues into it. One could go into laboratory genetics via having been in pathology, doing like clinical pathology, laboratory medicine. I actually started in pathology and did a year of pediatrics because I wasn't sure what I wanted, did the year of clinical genetics, and then did the time in the lab. It is possible to go straight to the lab. It's a LGG, or laboratory genetics and genomics, is a three-year course of study, a 3-year fellowship, and one could go straight into that. To be a clinical geneticist, I believe you have to have 2 years of training in something and 2 years of clinical genetics. So mine that was covered by my time in pathology and pediatrics and then time in genetics. But it is possible to go straight into the lab. In order to have further options like working in a clinic, to be able to work in a clinic, one would have to do some clinical time. If one wanted to be both a practicing physician and diagnostician. But I think it's completely reasonable to go straight into laboratory genetics and research straight out of medical school.
Okay. So given the hour, if anyone has any other questions, please write them down. But right now, what I'd just like to do is show you some of the available opportunities that exist for continuing your consideration of genetics as a career choice. These are a series of opportunities that we have gathered together, but they're out there. One is if you want to have a one-on-one conversation with a geneticist, and this could include a general or clinical geneticist, biochemical or metabolic, research or industry. It's a time for a one-on-one analysis of what might be your career path. Let us know, and we can put you in touch with a geneticist near you. It is useful if you prepare a letter of intent and provide a CV in advance to help focus your discussion. And that includes the geneticists that we have here tonight. If you want to visit the American Society for Human Genetics website, they have printed interviews with researchers who discuss their scientific papers, their career journeys, or their interesting teaching or public health initiatives. And this link will be provided to you in the extra opportunities brochure. If you're interested in doing an elective, a shadowing opportunity with a geneticist, hear about a patient, hear about a disease, the concerning issues of the day, these often require advanced planning. Let us know, and we can try and put you in touch with a geneticist near you. Again, prepare a letter of intent and a CV in advance, and that could help clear the way. We are at RNE pioneering the concept of an online elective, which is an online counterpart to the in-hospital experience where you shadow a clinician. We're going to review about 2 or 3 diseases and hear from patients with those diseases. The whole elective is about an hour to an hour and 15 minutes, and each elective has a particular theme. We have 2 scheduled, one on Wednesday, April 28th, and one on Tuesday, May 11th. If you're interested, let us know. RNE also has a Rare Disease Day speaker series where the Rare Disease Day is the last day in February. And so the Rare Disease Day season is like January and February, so we're just out of it now. But we presented in 11 venues across New England at Grand Rounds or some other presentation where we give a medical overview for 15 minutes, followed by patients telling their story. If you are interested, let us know, because many of the online remote programs are available. I know of at least 3 that we can get you links if you're interested in seeing what happened. One was at University of Vermont, and one was at University of New England, as well as Rhode Island University. And these are the disorders that were presented by the patients. And 2 more, the American College of Medical Genetics has a summer scholars program. Unfortunately, those applications for this year are all in, and you have to be a first-year medical student to apply. So here's the link, and it's an opportunity for next year. But there's also the Global Genes Rare Compassion Project, where accepted applicants who can be medical or healthcare students are matched with rare disease patients and families. The applications are accepted year-round and matches occur quarterly, and those who are matched are eligible to submit an essay to qualify for the Cox Scholarship of Rare Compassion Scholarship. So, and there's the link for that. So at that point, I'm going to stop sharing. Again, these opportunities will be provided to all applicants. We'll be listing those. In the meantime, I want to thank Lisa. I want to thank Jody. I want to thank David for sharing your time, sharing your passion, and helping to explain why this is a very formidable area of medicine to consider as a career choice. Again, folks, if you have any questions, don't hesitate to reach out. Sorry. One question here for us helpless kids fresh out of undergrad. What might this letter of intent look like? I've never done one before. You can write, just give us an indication of who you are, what you've done, and what you're interested in and why. That's all. And then we'll connect you. All right. It's a little past the hour. Thank you so much. If you have, follow us on Rare New England. That's www.rarenewengland.org, or write to us at info@rarenewengland.org and follow us. We have lots of good things going on. Thank you so much for attending tonight, and we'll see you soon.
Thank you, everyone.